So, you know the feeling. You have a terrible headache, or maybe your back is aching after a long day.
Yeah, we've all been there.
Right. So you reach into your medicine cabinet for a common over-the-counter painkiller, acetaminophen, Tylenol, basically.
Exactly.
And you swallow it without a second thought. You're expecting a very linear, very clean clinical outcome. The pain simply goes away. But what happens if you accidentally take just a little too much?
Well, it crosses the line into drug-induced liver injury, or DILI, which is actually a leading cause of acute liver failure worldwide.
Which is terrifying because it's so incredibly common.
It really is.
And normally, that's where the story ends. You took too much of a drug and your liver took the hit. But what's wild is this new paper we're looking at today. It shows that the rescue squad for your failing liver actually deploys from your gut.
Yeah, it's an amazing connection. We are looking at a newly discovered gut-liver axis.
Right. And the mission of this deep dive is to unpack exactly how that works. We're going to explore how a specific community of bacteria living in your intestines can remotely deploy chemical messengers to armor your liver against toxic drug overdoses.
It sounds like science fiction, honestly.
It really does. But to understand this rescue mission, we first have to understand the baseline problem. When you swallow that pill, it eventually makes its way to the liver to be processed, right?
Right. So in the liver, acetaminophen, which researchers usually refer to as ACP, is metabolized by specific cytochrome enzymes. The main ones are CYP2E1 and CYP1A2.
Yeah, CYP2E1 and CYP1A2.
Yeah, and their entire job is to process the drug. But in doing so, they convert the acetaminophen into a highly toxic, highly reactive compound called NAPQI.
Wait, so the very enzymes trying to process the painkiller are inadvertently creating a poison?
They are. But, evolution has a built-in backup plan. Normally, the liver rapidly detoxifies this NAPQI by binding it to a molecule called glutathione, or GSH.
Okay, glutathione.
Right. Think of GSH as a neutralizer. It grabs the toxic NAPQI, turns it into a harmless metabolite, and you just excrete it safely.
That makes perfect sense for a normal dose. But what exactly is the tipping point during an overdose?
It's purely an issue of volume. During an overdose, those cytochrome enzymes just go into overdrive.
Because there's so much of the drug.
Exactly. The production of toxic NAPQI absolutely skyrockets, and it completely depletes the liver's finite stores of protective glutathione.
Oh, wow. So you just run out of the neutralizer.
You do. And once that GSH is gone, the unneutralized NAPQI wreaks havoc. It binds to cellular proteins, triggering severe oxidative stress, especially in the mitochondria.
And what's the ultimate result of all that stress?
Hepatocyte necrosis. The liver cells quite literally die en masse.
Okay, let's use an analogy here to really lock this mechanism in. Imagine your liver is a vast recycling factory, right? And those CYP2E1 enzymes are the factory incinerators.
They like that.
So the acetaminophen is the incoming trash. Normally, the incinerators burn the trash and produce this highly toxic ash that's the NAPQI.
Right.
But you have a specialized team of safety workers, that's the glutathione. They immediately bag up the toxic ash so it could be safely disposed of.
That's a great way to visualize it.
Yeah, but during an overdose, thousands of trucks dump trash all at once. The incinerators run red hot, churning out mountains of toxic ash. Your safety workers are completely overwhelmed and run out of bags.
And so the toxic ash spills everywhere.
Exactly. And it literally burns the factory down.
That is precisely what happens at a cellular level. The mitochondria, the power plants of the cell fail. The endoplasmic reticulum undergoes massive stress. The cells burst and the liver shuts down.
So just to make sure I'm tracking the baseline here, let's do a quick recap. If I take too much Tylenol, my CYP2E1 enzymes process it into toxic NAPQI, which depletes my glutathione safety workers, and that toxic ash literally burns my liver cells to the ground.
That's the tragic baseline, yes.
So think about your own medicine cabinet for a second. Have you ever considered that processing your daily medications relies on a strictly finite supply of cellular safety workers?
It's a sobering thought. Once they're gone, you are entirely unprotected.
Or at least that's what we used to think. This is where the story takes a really fascinating turn into the microbiome, the gut bugs.
Yeah, the researchers stumbled onto this next clue almost by accident.
Oh, really? How so?
Well, they were studying wild-type mice, specifically a standard laboratory strain. But these mice were housed in two completely different animal facilities. Let's call them Facility A and Facility B.
Yeah, two different buildings.
Right. And when they exposed both groups to toxic doses of acetaminophen, the mice in Facility A survived significantly better than the mice in Facility B. They had much less liver damage.
Wait, if they're the exact same genetic strain of mice, why would the building they live in matter?
Because the environment dictated their baseline gut microbiota. Mice in Facility A naturally developed a different community of gut bacteria than the mice in Facility B.
Oh, that makes sense. Different building, different ambient bacteria.
Exactly. And to prove this was the deciding factor, the researchers performed fecal microbiota transplantations, or FMT.
So they took the poop from the survivors.
Right. They essentially took the gut bugs from the surviving Facility A mice and transplanted them into standard antibiotic-treated mice.
And what happened?
Those recipient mice suddenly gained the superpower of surviving the massive AP overdose.
Okay, I have to push back here. I'm a bit confused. The gut is essentially a tube running through the center of the body, right?
Yeah.
The liver is a totally separate, solid organ. How does the liver actually know what the gut bugs are doing? How does that survival signal physically bridge the gap?
That is the missing link. When researchers looked closely at the livers of the mice from Facility A, the survivors, they found a massive surge in the production of a specific molecule called SNHG9.
SNHG9. That's an RNA molecule, right? But not the standard kind.
Right. It's a long, non-coding RNA, or LNC RNA.
Non-coding, so it doesn't build proteins.
Exactly. Usually when we think of RNA, we think of messenger RNA, like a physical blueprint that tells the cell how to build a protein.
Yeah.
But non-coding RNA doesn't build anything.
So what does it do?
It acts like a cellular floor manager. It walks around the factory, organizing, directing, and turning other cellular machinery on or off.
So the liver is frantically producing this manager during an overdose.
Yeah.
Does this manager just stop the drug from turning into toxic ash?
No, the toxic NAPQI is still being made. The glutathione safety workers are still depleted. What SNHG9 does is intercept the downstream propagation of the injury.
So it stops the factory from burning down, even though the toxic ash is still spilling everywhere.
Precisely. And to prove this manager was doing the saving, researchers engineered mice with permanent extra SNHG9 just in their livers.
And what happened when they overdosed those engineered mice?
The protection was staggering. Blood markers for liver damage plummeted. When they looked at the tissue, they saw far fewer inflammatory immune cells swarming the area, and the liver cells had practically stopped self-destructing.
Wow. And this isn't just a quirky mouse thing, right? This translates directly to humans.
It does. The researchers examined liver biopsies from actual human patients suffering from drug-induced liver injury using fluorescence in situ hybridization, or FES.
Okay, FES. And what did they find?
When they mapped the RNA in those human tissues, SNHG9 was significantly higher in the DILI patients compared to healthy controls.
Which might sound backwards at first. If SNHG9 is high in damaged livers, is it causing the damage?
It's the exact opposite. It's a compensatory rescue response. It's the liver calling 911.
Oh, I see.
The human data proved this. Among the patients with liver injury, those who naturally had the highest SNHG9 levels actually had the lowest ALT and AST.
Those are the key blood markers for liver damage, right?
Exactly. So the more of this RNA manager your body produces, the better your liver protects itself.
Let's piece this together so far with another quick recap. Differences in our gut bacteria dictate whether our liver survives an overdose.
Right.
And this survival mechanism relies entirely on the liver upregulating this non-coding RNA manager, SNHG9, to halt the damage. Human data proves high SNHG9 correlates with less severe damage.
Spot on.
It makes you wonder, if your liver has a hidden emergency manager like SNHG9 that only clocks in during a crisis, what other invisible cellular managers might be operating in your tissues right now to keep you safe?
It's a profound question. But to answer how this specific manager actually pulls off the rescue, we have to look at the exact micromachinery it controls. We need to zoom in even further.
Yeah, let's unpack that.
Yeah.
Because you can't just throw an RNA molecule at a dying liver and hope for the best.
Yeah.
How is a manager molecule actually putting out the fire?
It's a highly precise intracellular chain reaction. SNHG9 operates by physically binding to a specific protein called IMP2.
Okay, so SNHG9 grabs onto IMP2.
Right. Researchers proved this via RNA pull-down assays, and they specifically pinpointed that the binding happens at loop 3 of the SNHG9 structure.
Loop 3. Very specific.
It is. And when it binds, it forces IMP2 to interact with the messenger RNA of another gene called MYC. But this interaction is hostile.
Hostile how?
It destabilizes the MYC mRNA, which completely suppresses the translation of the MYC protein.
Why do we want to suppress MYC?
Because MYC acts as a genetic repressor, essentially a brake pedal on a crucial, highly protective gene called MAS1.
Oh, I see. So by binding IMP2 and destroying MYC, SNHG9 is taking the brakes off MAS1. It's suppressing the suppressor.
Precisely. With the MYC brake removed, MAS1 expression skyrockets. MAS is a G protein-coupled receptor, and its activation initiates a massive cellular cleanup operation.
How does it do that?
Biochemically, MAS activation shifts specific signals. We see a decrease in AKT phosphorylation and an increase in FLXO1 acetylation.
Okay, a decrease in AKT phosphorylation and an increase in FSO1 acetylation. What does that chemical shift actually trigger in the cell?
It acts as the ultimate green light for a process called autophagy, specifically mitophagy. It upregulates genes like ULK1, ATG7, and LMP1.
Autophagy, that's when a cell eats itself, right?
Basically, yes. It switches on the genes responsible for building autophagic vacuoles, which are basically cellular trash compactors.
So because the toxic acetaminophen ash is destroying the liver's mitochondria, the MAS receptor tells the cell to literally eat its own damaged, burning mitochondria to prevent them from poisoning the rest of the cell.
That's exactly it. The cell digests its dying quartz to survive.
That is incredible.
Researchers proved this by knocking out SNHG9 in human liver cells. When they did, cellular trash rapidly piled up because the autophagy cleanup process was broken.
And when they put it back?
When they added SNHG9 back, the recycling resumed and the cells stabilized.
Okay, there's an easy way to remember this cascade. If your liver is stressed, this pathway forces it to simmer down.
Simmer down.
Yeah, SIMA. SNHG9 binds IMP2, which muffles MYC, which activates MAS, leading to autophagy, the simmer down pathway.
I love that. That's a great way to map it. And they saw this exact cascade active in the human liver biopsies. The high SNHG9 levels were perfectly matched with elevated markers of active cellular recycling.
So to summarize this rescue mission, SNHG9 binds IMP2 at loop 3, suppressing MYC. That takes the brakes off MAS. And MAS triggers autophagy via that AKT and FLXO1 signaling to clear out the toxic debris.
You've got it.
Can you picture your liver cells literally digesting their own damaged parts just to survive a drug overdose? It is the ultimate form of biological recycling.
It really is.
But, this leaves the biggest mystery hanging. We know how the liver saves itself. But how did the liver know to start the simmer down pathway in the first place? What was the signal from the gut?
To find that signal, researchers had to go back to the feces of the surviving mice from Facility A.
Right, the 16S sequencing data.
Exactly. They sequenced the bacteria to find out exactly who was living there, and the true bacterial culprit, the hero, turned out to be a specific probiotic strain called Lactobacillus plantarum.
Lactobacillus plantarum. So when they gave mice this specific bacteria, it protected them.
Profoundly. Mice given oral Lactobacillus plantarum showed massive upregulation of that liver manager SNHG9 and vastly improved survival against the acetaminophen overdose.
But as I pointed out earlier, the gut is a separate tube. The bacteria stay in the intestine. They don't migrate to the liver.
Right, so they have to be sending a chemical messenger. A text message, if you will.
And how do they prove that?
To prove this, the researchers took the supernatant, the liquid that the bacteria were grown in, which contains all their chemical secretions, but they removed the actual bacteria.
So just the bacterial bathwater, basically.
Exactly. Only the liquid was given to the mice, and the liquid alone protected them. The text message was in the secretion.
And what was the message? What's the specific chemical?
It's a microbial metabolite called HMB, 2-hydroxy-2-methylbutyric acid.
Wait a minute, HMB. Isn't that the popular supplement that bodybuilders take for muscle recovery?
It is. It's the exact same molecule.
That's wild.
HMB is naturally derived from the essential amino acid leucine. Humans can synthesize small amounts of it, but it turns out our gut microbes, specifically Lactobacillus, produce it too.
So athletes take it for muscle recovery, but here the gut bacteria are using it for a totally different function. How does HMB physically talk to the liver?
The HMB travels from the gut through the blood directly to the liver. And on the surface of the liver cells, there's a specific receptor called HCAR2.
HCAR2, hydroxycarboxylic acid receptor 2.
You got it. It's a Gi-coupled receptor. And researchers proved HMB binds to it by showing a resulting decrease in CAN-AP levels inside the cell.
So HMB locks into HCAR2. Is that what it finally turns on the emergency manager, SNHG9?
Yes. That exact binding event commands the liver cell's nucleus to start manufacturing SNHG9. To absolutely prove this, they gave the mice a drug that chemically blocked the HCR2 receptor.
And if the receptor is blocked, the text message can't be received.
Precisely. When HCR2 was blocked, the HMB did nothing. SNHG9 wasn't produced, the simmer down pathway failed, and the liver suffered massive necrosis from the drug overdose.
Wow. And does this bodybuilding metabolite hold up in the human clinical data?
It perfectly mirrors the mouse data. In human patients suffering from DILI, blood tests revealed a perfect negative correlation.
Meaning what, exactly?
The higher the patient's serum HMB levels, the lower their ALT and AST liver damage markers. The more HMB circulating in their blood, the safer their liver was.
So closing the loop on this segment, the gut bacterium Lactobacillus plantarum secretes the metabolite HMB. HMB travels through the blood and activates the HCAR2 receptor on the liver.
Right.
And that HCAR2 activation is the master switch that turns on the protective SNHG9 cascade.
Beautifully summarized.
Does knowing that a specific molecule produced by a specific gut bacterium can physically intercept liver failure change how you view your daily diet and gut health? It's not just about digestion, it's about systemic armor.
It really is a whole-body defense system.
Let's do a quick, rapid-fire grand synthesis of this entire pathway for the listener.
Let's do it.
You take too much Tylenol, your liver starts to die as enzymes create that toxic NAPQI.
But waiting in your gut is Lactobacillus plantarum. It senses the environment and releases the metabolite HMB.
The HMB travels through your blood to the liver and hits the HCAR2 receptor.
The liver responds by frantically churning out the non-coding RNA manager SNHG9.
SNHG9 springs into action, binding to IMP2. This shuts down MYC.
With MYC gone, the MAS receptor activates. This kicks off autophagy.
The cellular trash compactors turn on, clearing out the toxic mitochondria, and your liver lives to fight another day.
From a bacterial secretion in the intestine to a genetic rescue mission in the liver. It's elegant.
It really is. So here's a final forward-looking question for you to mull over. If our microbiome naturally produces protective molecules like HMB to combat drug toxicity, are we approaching a future where doctors don't just prescribe a pill?
That's the million-dollar question.
Right. Are we entering an era where doctors prescribe a specific tailored probiotic alongside every potentially toxic medication just to preemptively armor your organs before the damage even starts?
It completely reframes how we view drug safety and the microbiome's role in it. The bacteria aren't just along for the ride. They are active participants in how we process medicine.
Think about that the next time you reach into your medicine cabinet for a simple headache pill. You aren't just treating a symptom. You're initiating a massive, invisible biochemical conversation between your bacteria, your RNA, and your organs. Thank you for joining us on this deep dive into the hidden machinery keeping you alive. Until next time, stay curious.